Science held together with masking tape. Hey everyone, my name is Chris Anderson. Cincinnati Children's is one of the best hospitals in the entire country, but it's also home to some world-class research. Dr. Richard Liu is a geneticist here, and he and his lab have made some amazing discoveries about brain diseases, like multiple sclerosis and cancer. I can't wait to find out more about what they're learning.
So my name is Richard Liu. I'm a professor and the scientific director of Brain Tumor Center at the Cincinnati Children's Hospital Medical Center. I always fascinated by the nature because I grew up in a rural area, so I was always wondering how things work around me. When I was in high school, so I found out I can see the chemical reaction in the chemistry textbook can really happen in the test tube. So and then I said, wow, this is really true. So this is a wow moment for me. I mean, so my interest in science soared.
Dr. Liu.
Hi, Chris.
Good to see you.
Good to see you.
So, I have a question for you. There's lots of different types of nerve cells. What kind of nerve cells are you and your lab researching?
I see. So we are particularly interested in one of special cell type in our brain. It's called oligodendrocyte.
Oligodendrocyte?
Oligodendrocyte.
Oligodendrocyte. Okay, so
Right.
What's so special about these cells?
So oligodendrocyte is the cells which can produce myelin sheaths.
Myelin sheaths.
Right. So they can wrap around the axon neuron.
And what's so special about that myelin sheath?
Right. The myelin sheath is just like the insulation for electrical wires.
Okay.
Right. So without that insulation, there's no the neuron cannot function.
Okay.
Right.
So these cells make the insulation for our nerve cells.
Yes.
So they can transmit all sorts of signals between our brains and our bodies, right?
Yes.
So what happens when these oligodendrocytes, what happens when they don't work or they stop functioning?
So if those cells cannot produce a myelin sheath, so our neuron is not functional. So therefore we cannot communicate each other.
So you lose brain function.
Yes. Right.
So what kind of diseases can happen when the myelin sheath isn't working?
There are many kind of disease. The most famous one is the multiple sclerosis.
Multiple sclerosis.
Right. So we try to strive to identify those genes can promote the differentiation process.
Okay.
And then to push those cells to start making myelin sheaths.
So you guys are looking at the genes that turn these cells on and get them making myelin sheaths again.
Yes.
So you guys could really help people with MS or brain cancer.
Yes.
Can you show me more? Can you show me more about your work?
Oh yes. So I can show you a run.
Awesome.
Right.
Awesome. So Dr. Liu, what are we looking at here?
So we are looking for in the brain section. This brain section coming from the mouse brain.
Okay.
And this mouse actually developed brain tumors. You can
So
see
this is a brain tumor.
brain tumors.
In one of the animals that you test.
Yes.
So
right.
this is it right here.
Yeah.
So why is this a darker color? Why is this a different color than the other parts
Because
of the brain?
the brain tumor cell tissue, they have very high cell density. So they
Very
look
dense, a lot
very
of cells
dense,
in a small area.
In a small area.
Okay.
Right.
So that's why that's showing up differently.
Right.
So do we know why this mouse that has brain cancer here?
So we can develop an animal model to model the human brain tumors because we know in human we have a genetic mutation, particularly in the tumor suppressor genes. So we can recapitulate the phenotype in a mouse model.
So you guys are, let's get this straight. So you guys are looking at the genes that turn on and off brain cancer.
Yes.
And you guys can look at the genes in this mouse?
So we can look at we can make a mutation. For example, we can use CRISPR.
So you guys are using CRISPR to do
Right,
this?
yes.
So you guys are changing the gene to see if it turns on the brain tumors.
Right. So we know in the human there's a faulty gene can cause the brain tumor formation.
Sure.
So therefore we can use CRISPR to mutate that gene.
And do the same thing
do
in
the
mice.
same thing in the mice.
That's crazy. So you guys are turning on and off genes.
Yes.
That's awesome. What have you learned from this that we can use with humans?
Yeah, so by establishing mouse animal model for brain tumor, so we can look at how tumor cell develop, and we can also look at what's the cell origin for the tumors.
Okay.
And then with this system, we can treat the animal with drugs we identify from tissue culture.
Okay.
And then treat those drugs treat the animal with the drugs, and then see how this drug function to block the tumor.
And if the mouse and the mice get better, then you know that could work for humans.
Exactly.
That's amazing.
Right.
That's so cool.
Yeah.
I mean that's just cool. So how are you guys able to change the gene? You said with CRISPR. How does that work? How are you guys able to do that?
Yeah. So normally you design a RNA called guide RNA.
Okay, so they've got like a little piece of RNA that guides.
Right, they can target to the genome specific segment.
Okay, so you have this little guide RNA that'll match up right with the gene that you want.
Right,
okay.
And then you can you can delete the gene, you can target the deletion, or you can reverse the faulty gene into the normal one.
Okay, and you guys can just do it here with like pipettes?
Yes. So we can generate this guide RNA, and then we can inject them into the cell or into the mouse brain.
Just right in the mouse brain.
Yes.
And it will change the genes so you guys can look and see the effects of that.
Yes.
And you guys just do it right here, like with these pipettes?
Yes. Yeah. It's very simple. Everybody can do it.
Well, anyone can do it. Yeah. Anyone can research brain genetics. You can do it at home. So Richard, what else do you want to understand about the brain?
So next is try to understand how brain function and how can we help to restore the damaged nerve system.
So how can that help people with, say, MS or brain cancer?
Recently, actually, we identify a drug can help, potentially help to promote the myelination process. So this kind of drug function also can prevent the cell overgrowth, to prevent tumor growth.
So this drug can not only help the myelin sheath to regenerate, to have those oligodendrites regrow that myelin sheath, but it can also keep brain cells from overgrowing.
Yes.
And keeping people from getting brain cancer.
Yes. It's more like the one stone to kill two birds.
That's awesome.
Yeah.
Well, Richard, thank you so much for inviting me into your lab and sharing your awesome work.
Thank you, Chris.
Yeah, absolutely. And thank you for Children's Hospital for allowing us to tell Dr. Liu's story and the work that they're doing here. And thank you for watching, and we will see you next time on Science Around Cincy. Dr. Richard Liu and his lab have made some amazing discoveries about brain. Some amazing things. Sorry. I can't wait to learn more about what they're discovering. I'm gonna have to do that again because that car. Yeah. Cincinnati Children's is home.
Yeah.
Yeah.
Cool.
Cool.